Oh, there we go. Hi, welcome back. I hope everyone has enjoyed the morning. We're on a new StreamYard link now, but it's been a wild ride so far with so much new information. And I am so excited to welcome Kate Bemis as our next speaker. Now, Kate is a research zoologist at NOAA National Systematics Lab and the curator of fishes at the Smithsonian, a new post. And I am I love about Kate and the chapter that she lead authored is that she did it with her dad, Willie Bemis, who is a famous fish morphologist that I studied and I studied about in graduate school. And it's just so, I love that it was this father-daughter team coming together. And some of the discoveries that you've made in taking a deep dive into mola anatomy are just phenomenal. So I can't wait to hear your talk.
Great. Thanks so much, Tierney. Can you see my slides?
Yep, you're good to go, Kate.
All right, great. Thanks, Joe. Great. Well, I'm just having such a great morning learning about ocean sunfishes from all the wonderful talks that have been given so far. And like Tierney said, I'm a research zoologist at the NOAA National Systematics Lab and curator of fishes at the National Museum of Natural History in Washington, D.C. And molids are, of course, some of my favorite fishes, particularly because they have some really unusual anatomy that we'll talk about today. And on the slide right now, I'm showing ontogenetic series, which have come up a lot already today, of Ranzania in the top row here, and then Masturus in the middle, and on the bottom, Mola, the ocean sunfish. So before I get started, I just want to say a quick thank you to some folks, especially the organizers of the book and the symposium, Tierney, Graham, and Jonathan. And as some background, it was in September of 2018 that I first learned that this book was being put together when I was a grad student at the Virginia Institute of Marine Science. And Jim Tyler, who I had recently collaborated with, encouraged me to get involved in this book. I'm so grateful for my PhD advisor, Eric Hilton, who, when I asked what he thought about this crazy idea, he said, of course, go for it. And like Tierney mentioned, I also asked my dad, Willie Bemis, to join the team. And here are the four of us in one of doing a dissection of Masturus late last year. So again, I just want to say thank you so much, Tierney, for getting this organized. This project took me in so many directions that I never would have gone in had you not organized this symposium and book volume. So the anatomy chapter that we prepared for the book is a review based on existing literature. And today what I want to talk about is some of our new findings based on our own dissections and CT scans that fit and really were guided by that review chapter that's in the book. So of course, we're going to talk about Ranzania laevis and Masturus lanceolatus, and then all the observations I'm going to show today are just on mola. So there's still lots of exciting things to do with the other species of mola and their anatomy. So just to get us started thinking about the anatomy of molids, there are really two themes that explain many of the unusual features that we see. And the first is anatomical reduction. So relative to other tetradontoids, in molids we see that they have a reduced skeleton. Molids also do not have a caudal peduncle or caudal fin. Of course, it becomes replaced with the clavus. They've lost axial muscles. The stomach is simplified into this eye-shaped that runs directly from the esophagus to the intestine, and it's not really demarcated by any valves. Molids have a really short spinal cord, and it's actually totally contained within the brain case. In terms of their sensory systems, at least 3 of these are really reduced, including that they have tiny, poorly consolidated otoliths. They also have tiny nostrils, which lie nearly flat against their skin and have a almost flat olfactory epithelium and no nasal rosettes present. In terms of their lateral line, molids don't have any canal structures, and all the neuromass that they do have are superficial. And the lateral line is contained only on the anterior part of their body. And finally, we see that in adult sunfishes, they don't have a swim bladder. But perhaps even more striking than these losses and reductions are the unusual anatomical specializations that molids have come up with. And that, of course, most famously includes the clavus, which is formed from dorsal and anal fins. If you've dissected specimens of ocean sunfishes, you know that the bone of molids feels more like cartilage than it does like true bone, which we attribute to this distinctive honeycomb-like pattern that we see in the histology of it. Molids have a greatly thickened hypodermis, this white tissue here, and they also have really unusual scales. At least in Mola, they have these highly modified scales that are very branched and almost hair-like at their tips. There's a mesenteric tissue that surrounds the intestines, holding them together. They also have a really large heart that has anatomical features that are unknown in other fishes, such as having 4 leaflets in the valves between both of their chambers. And this specialized heart pumps blood over a really large array of gill filaments that are supported by novel structures. And finally, the kidneys are also really large in ocean sunfishes. So it's together that these reductions and specializations combine into forming these really unusual fishes that we love for their anatomy. So as you can see from these illustrations that are on the screen now that were summarized by Steenstrup and Lutkin in 1898, molids, perhaps because of their unusual anatomy, have really fascinated people for nearly 500 years. And so there's a really rich literature of anatomical data and interpretations about their anatomy. Accounts prior to the 21st century describe molids as being slow, inactive fishes, even so much as describing them as sleeping on the surface, based on observations of molids basking. I just have to read you one of my favorite quotes from Burr in 1928, who wrote that its habits are described as being lethargic, slow-moving, not infrequently found apparently asleep on the surface of the water. But we now know from tagging studies that I imagine we're going to hear even more about this afternoon from folks who actually did these tagging studies that are in the symposium, that ocean sunfishes are actually quite fast and that they can swim at speeds regularly of up to 0.5 meters a second, which is comparable to other large fishes and pelagic sharks. And it's not just that they're going fast, but they're also making deep foraging dives, typically to 150 meters, but at least as deep as 800 meters. And they're doing this to target siphonophores and jellyfishes, among other gelatinous prey. So here are 3 frames that this was taken from a study by Nakamura et al. In 2015, where they attached a camera to the back of an ocean sunfish, and you're looking at the anterior part of the sunfish here and a jellyfish that has come into view. In the second frame, you can see that jellyfish has been sucked into the mouth of the ocean sunfish, and in the third frame has been released. And I show this because even though there's lots of data, or there's lots of literature on ocean sunfishes feeding on jellyfishes, it was really this study, I think, in 2015 that convinced me that this is what they're doing, that they're targeting ocean sunfish, or that they're targeting jellyfishes. So today, I think every organ system deserves comment because there's so many extreme examples in vertebrate morphology in ocean sunfishes. But I really want to focus today on just 3 examples that relate to the new data on the swimming capabilities of ocean sunfishes and also the new information we have about their diet. And I want to start by talking about teeth, which are some of my favorite anatomical structures. So in this slide here, I'm showing you, this is CT data, and on the left side here is the porcupine fish, which is a close relative of ocean sunfishes. And like the porcupine fish, the ocean sunfish has this beak that runs along both their upper and lower jaws. And in this view, this is an anterior view, and this is a view looking down on the surface of the lower jaw. So they have that beak in common with porcupine fishes, but posterior to the beak in porcupine fishes is this really large crushing surface that they use to process hard-shelled prey. And it's probably not surprising to you that when we look at the adult surface in ocean sunfishes, we don't see any of those crushing teeth. And this seems to fit well with what we know about what they're feeding on as adults, jellyfishes. But because our study was really interested, we were focused on looking at the ontogeny of ocean sunfishes. We also did some CT scans of juveniles and larvae. And this is just one scan of a larval sunfish. Not a larval sunfish, a juvenile sunfish, probably about this size. And right now, let me go back to the beginning. Right now, we're looking at it directly in anterior view, and you can see this upper beak that overlays the beak in the lower jaw. But if I play this video for you and we turn this sunfish around so that now, as it finishes here, we're looking out the mouth of the ocean sunfish, you can see that there are individual teeth that we don't see in the adult of this species. So what's going on here? To better understand how teeth change in ocean sunfishes, we CT scanned an ontogenetic series. And what we found is that early in their development, they have a beak that's present, but no individual teeth. And as they get larger, they have individual teeth that are paired that are posterior to the beak here. And these teeth continue to be present and actually expand along the jaw of ocean sunfishes until they start to disappear. And by the time we look at adult specimens, we see that they have no individual teeth in the region posterior to the beak. I think this is really interesting to compare to the porcupine fish because what we found in them is that they also have individual paired teeth early in their development that then they expand posteriorly and then start to develop on top of each other. And those teeth eventually form this large crushing surface or triturating surface that we see in adults. So there's a lot of similarity in their early ontogeny, but then they diverge into these two different structures in the adult forms. And at least we're starting to get more diet data for ocean sunfishes. And I don't want to steal Natasha Phillips' discussion of this in her talk later this afternoon, but I just want to put this up to show that there's now, especially by her work, showing that there are ontogenic diet changes in ocean sunfishes, and that early in their ontogeny, they are feeding on relatively harder organisms. And so perhaps these teeth that they do have at smaller sizes is related to what we see them feeding on. And by the time they get to be adults, they have diets entirely composed of gelatinous prey. All right, so now I want to switch to a different topic, and I want to talk about the swim bladder. So as background, all Tetraodontiformes, the group that Molids belongs to more generally, have a swim bladder except for one species of pufferfish. And all of the ocean sunfishes as adults lack a swim bladder. However, when we were reviewing the literature for this project, we found that Fraser Brunner in 1951 had described a swim bladder from a specimen of Masturis that was about 21 millimeters long. And since that description, the presence of a swim bladder in Molids has not been further investigated or included in other accounts. And so we were really excited to jump into this question to see if we could confirm Fraser Brunner's observation and also where else we see them in ocean sunfishes. And so this is a dissection of a 57 millimeter total length specimen of Masturis. And to orient you, here is the vertebral column along here, and the intestines and the liver. And we were so excited when we were able to find that there is in fact a swim bladder in this specimen. So from there, we wondered, well, what about Mola? What about Ranzania? Do they have swim bladders? And so we used micro CT scans to study really small individuals. This particular Mola is one of my favorite specimens. It's about 7 millimeters long, size of a pea. And we were able to digitally dissect this specimen. And what I'm showing on the screen now is in yellow is the gut tube of ocean sunfish here, and in green is the swim bladder. So this was super exciting because this hasn't been shown before. What it says is that there's this ontogenetic loss of this structure that they have, at least Masturis and Mola have as larvae and juveniles. And then as they get to be adults, they lose the swim bladder. And I think that this makes a lot of sense given what we now know about the diving behavior of ocean sunfishes, because if you're making repeated dives deep, you don't want to have an air-filled space because the changes in pressure are going to make that hard to maintain. So again, I would love to talk more about, where does this potentially suggest that ocean sunfishes as larvae and juveniles are spending their time that's different from what we know about the adults. Okay, so I want to tell one last story about anatomy today, and that's about the heart and circulation system in ocean sunfishes. So we now know that they're not the sluggish animals that they had previously been described to be, but we're still trying to understand how they do that. And one way I think might be their unusual heart. So on the screen here, this is a section of Masturis that we did, and you can see that they have, like other typical fishes, have a two-chambered heart that consists of the atrium and the ventricle. And then in ocean sunfishes, they also have the bulbus is much larger than in other species. So we took this heart and we cut it in half. This is a sagittal section. And to orient you, I've labeled the atrium here and the ventricle, and then here's the bulbus. And through this heart, deoxygenated blood moves along this path and then goes out to the gills. So the last person to talk about hearts of molids in any detail was Rosen in 1913. And one unusual feature that he tentatively described at that time was that there are 4 leaflets in the valve between the atrium and the ventricle. So that atrioventricular valve, we were able to re-examine in fresh specimens, and you can see the leaflets here that has this stiffer yellow tissue that stands off of the wall of the heart. So that was really exciting to see. But it's not just that valve that's unusual. The valve between the ventricle and the bulbus also has 4 leaflets, and you can see them labeled here. And in both cases, this is more leaflets than are known in other fishes. And likely what that means is that it results in more complete closure of the orifice between the chambers, which would limit backflow during contraction. There's some other really crazy things going on with their circulatory system. One I'll just mention is that they have 3 distinct coronary arteries, whereas other fishes typically only have 2. And together, all of these observations about the heart suggest that ocean sunfishes have a powerful circulatory system. And the reason you would want to have a big heart is because you need to drive blood through an extensive series of gills. And the gills of ocean sunfishes are also unusual because they're surrounded by this tissue or what we call the gill sac. And so if you open up the gill sac, we can see this great expansion of the gills. And it's actually only the gill filaments that are in this middle region here, which was first described by Adney and Hughes in 1977. Only the gill filaments in that region are supported by skeletal structures that typically support gill arches, like the ceratobranchioles, which are some of the bones in the gill arch. And it's the expansion that occurs in this area here and also above the middle region are only possible because they have, ocean sunfishes have really densely mineralized gill rays, which are the structures that support the filaments. And then at the base of those gill rays, there are novel cartilaginous supports that we don't see in other fishes. So I think that it's likely that there's some relationship between the powerful heart and the gills of molids and what we see them being able to do in the water in terms of their speed. But there's still so much to learn about circulation and respiration in ocean sunfishes. And I would love to see, now that we have these hypotheses about their anatomy, to restudy some of the physiology to better understand the metabolism of molids. So I hope what I've showed you today is that even with nearly 500 years of study, we're still learning so many new things about ocean sunfishes, in particular in relation to this new ecological and behavioral data that's now available. And I want to say thank you to everyone who's been engaged in this project. This has been a really big effort to borrow specimens from natural history collections around the world and folks who collected new specimens in the field for our work. And again, thank you to all the organizers and my co-authors for helping to make this project happen. Thanks.
Hey, it's so completely fascinating, what you've uncovered. I mean, and just such beautiful dissections. I know it is not easy, especially with samples that have been sitting in formaldehyde and ethanol for many decades, to pull out these things that you have found. It is really, I mean, just such gorgeous work. You and Willie you're just masters.
We had a really good team of folks working on this, and it was just so much fun to learn about sunfishes with Jim and with Eric and my dad. And I'm so glad you got us started on this path.
Well, and the work you're doing, I mean, the CT imagery and the spinning around to look down, I mean, spectacular that you can now see into these areas, and the swim bladder work. Were you able to see if Ranzania had a swim bladder?
Yeah, so that's a great question. I did look for a swim bladder in small Ranzania that are, probably in this size range, and I didn't find one. And that doesn't mean it's not there. The swim bladder in all small ocean sunfishes has a really thin wall, and so it's certainly possible that they do have one, but I haven't seen any evidence of one in my dissections.
And you haven't micro CT'd any Ranzania larvae?
We have, but it has to be, in order to study the soft anatomy using CT, you either have to stain the specimens so that you can have better contrast between the soft tissues and the bones. And I have not done that for Ranzania in a very formal way.
Yeah, it'll be so interesting because, what's going on with the Molicanthus and if they don't have a swim bladder functionally. Yeah, absolutely. And wouldn't it, what I would do to put them in a flow tank and do the kind of work like what Barbara Block has done looking at hearts and tunas.
I would love that so much.
There's a lot of heart physiology and just looking at the heart muscle and enzymes, and there's a lot of really interesting questions that could be asked just physiologically with the heart and the sunfish.
Yeah, I absolutely agree.
Yeah.
I'm going to be a good ex-PhD supervisor, Natasha, and not ask the 37,000 questions that I really want to ask you. But I would ask if you guys can hook up and chat afterwards, because there's so many good questions about ontogeny. Lesson from your talk, which I like, is always say yes to the weird random invite that you just don't know if you should.
Yeah, I learned that lesson too here.
It's always good to do that. And I also loved, because it's cool, you kind of got into sunfish in a way, sort of like me and Tom got into lots of weird things. I think there's a really healthy period where you follow your gut more than the literature, and you're cynical and you're like, I don't know. And I think when you're saying I didn't really buy into this idea of, sunfish eating jellies, I agree. They're rubbish at doing it. If you actually see them trying to eat it, that whole that jaw structure. And I think people, it was just that constant kind of reiteration of, the obligate predators of jellyfish.
Yeah.
I know that when they're small, we should know that their diet is heavily, kind of benthically driven. They've got a raspy parrot flipping fish beak thing going on. It makes sense. If you eat jellyfish, they kind of eat them and then go and spit it out again, and it looks weird. So that's like the mechanical explanation of what we've kind of knew what was going on, but it's just so those images were stunning. Absolutely gorgeous. So
really looking forward to connecting with Natasha and talking more about changes in diet and all things sunfishes.
Man, I could go on, but yeah. That's put the ball on the tee. That's beautiful. Thank you. I'll shut up. I promise to.
And Mayana too. Definitely got to connect with Mayana with looking at these larvae has been because there's there's just so many secrets that can be. We would have, had you really looked closely at the teeth early on, you would have seen that there must be some sort of ontogenetic shift because there's such a radical change in their jaw. I love your comparison with Diodon. They just really build up that triturating surface, and the molas are like, we're done with it.
We don't need that anymore.
We're done with that. So it's I just love how anatomy can lead you to functionality, And anytime like my advisor, Steve Wainwright, would say, nature reveals her great secrets in her extreme forms. And the Mola is absolutely a case study in that. So
absolutely.
Can't wait to keep following all the new dissections you have, Kate.
Me too. I hope we get back in the lab at some point soon to do a couple more, and then we'll wrap up this part of ocean sunfish anatomy, and we'll find the next part to work on.
Yeah. So you do have that monograph that you're working on, yeah?
Yep.
And
yep.
When's that due to come out?
Well, it has to get submitted first, but hopefully that will happen later this spring, and then we'll go from there.
Yeah, because that's we're waiting for that with bated breath. That's going to be great. I'm sure you'll get the cover. Well, it's a monograph. You'll have the whole thing.
Yeah, I'll have the whole thing to myself.
All right. Well,
thank you so much for tuning in, John.
Give my best to your dad.
Pleasure. That was great. Thank you so much.
Bye.
Yeah.